In this section, we're going to look at control of the cell cycle, and your learning outcomes here are to distinguish the role of checkpoints in the control of the cell cycle. So there's going to be three main checkpoints. You're going to need to be familiar with what's being checked at those points. And then you're going to look at the role of a specific checkpoint and the molecules involved in control of that checkpoint, and this one is called anaphase promoting complex. It goes by the name called cyclosome. And so we start by looking at two concepts that are related. The first is that in the cell cycle, there's going to be two irreversible points in that cell cycle. One is going to be when you start to replicate genetic material. Remember that that's going to happen during the S part of interphase during the cell cycle. Once you commit to making your copies of your DNA, you can't stop that process. The other one is during anaphase. So the first one was the S phase, and the other one is anaphase, which is part of mitosis, and that's when the sister chromatids are going to be separated. Once they start to separate, you can't reverse that. The second major idea here is that you can put these processes on hold. Once you start these processes, you can't stop them. So you can put these processes on hold at certain checkpoints in that cell cycle. And these checkpoints are important because they're going to check the process to make sure that things that are supposed to have happened before progressing in the cell cycle have been done correctly or accurately. And if not, then we can stop and try to fix things before progressing. The other part to this is that the cell at these checkpoints is responding to both internal conditions as well as external signals. Cells respond to their environment, and in this case, the cells need to be responding to certain cues in the environment that may trigger them to progress through mitosis and so on. So the mechanisms have become understood through different areas of research and combining all of that information to better understanding of what's going on in control of the cell cycle. The first important research that was done on frog oocytes. These oocytes are cells that are going to go through a process called meiosis. It's kind of like mitosis, but in meiosis, you start with special cells that are going to go through divisions to form egg or sperm cells, which are going to be haploid in animals. Okay? And that process is called meiosis. So you get those haploid egg cells. So oocytes actually become eggs. Spermatocytes will become sperm cells. But they were looking at frog eggs, and these frog eggs are going to, before they go through meiosis, they're going to stay arrested in G2. And that means they don't start the division, because at G2, you're right before mitosis during the cycle of the cell going through division, but it's a special division called meiosis, but they're going to be arrested at G2. And so they had these frog eggs that were paused at that point there. So it's actually going to become one of those checkpoints. But what they did was they looked at other oocytes that were already progressing into meiosis. They were going through the division. And so what they did was they took the cytoplasm from the cells that were going through cell division, and they injected it into the oocytes that were arrested, that were on pause. And when they injected that cytoplasm in there, those oocytes began to go through meiosis as well. So there had to be something in there that was causing this, and they gave it a name called MPF. And MPF stands for maturation promoting factor. Some protein or something in there was causing the cell to progress. They also figured out that this MPF had an enzymatic activity. It acted as an enzyme, and the specific activity was to phosphorylate. Remember, to phosphorylate means you're going to take a phosphate and attach it to another molecule. In this case, this MPF was known to phosphorylate other proteins. And when you phosphorylate other proteins, that changes the activity of that protein. It can either activate it or inactivate it. So this MPF was involved in activating other proteins by phosphorylating. Whenever you have enzymes in general that do phosphorylation, they're called kinases. And so MPF was acting as a kinase. And so Then they looked at other researchers were looking at sea urchin embryos. And sea urchins are related to starfish. They're kind of animals that live in the ocean. And they discovered that in these that there were proteins that increased in their levels inside the cell in concentration and then decreased at regular intervals during the cell cycle. So in synchrony with the cell cycle. So it seems that every time a cell was going to progress and begin another stage in the cell cycle, we saw an increase in these proteins. They named these proteins, they called them cyclins. And they saw that there were two forms within the sea urchin embryos. One form peaked at before going into S, so at the G1S boundary. So you're in G1 of the cell cycle, and then S, remember, is where you're going to make your copies of your DNA. So the cyclins appeared to increase in level there. And another one was at the G2M. So at G2 is right before you go into mitosis. So right there you saw an increase in another kind of cyclin. So it would increase at that point and the cell would progress through there. So then another area of, you have in the frog eggs, they learned about an enzyme that phosphorylated. They called it maturation promoting factor because it causes the maturing of eggs, right? And then you have cyclins in the sea urchin embryos. And now they're looking at yeasts, and they're looking at mutants of yeast that could not progress through the cell cycle. So there's a mutation that caused them not to progress through the cell cycle. And so it's when you start looking at mutants that you start to learn what kinds of proteins those mutants are making, and when you compare them to those that are not mutants. And so in these yeast mutants, they found that they were not able to progress through the cell cycle. And in the yeast, there was going to be two critical control points. And one happened to coincide with what we saw with the cyclins in the sea urchin embryos. One was the synthesis of DNA. Remember, the synthesis occurs at S. So here we're talking about going from G1 to S. And the other one was the commitment to mitosis. So there you're talking about going from G2 to M phase, right? And the gene, so if the gene was mutated, we couldn't progress in there. And so they identified the gene that was associated with making the protein or the factors involved in progressing through there, and they called it the CDC2 gene. So the CDC2 gene is responsible for making a protein like any gene, and in this case, the CDC2 gene was responsible for making a kinase that we've been talking about. Okay, so it turns out that when they started looking at that CDC2 gene, as I mentioned, it was a kinase. It's an enzyme, a protein that phosphorylates. And they found that the MPF that they were looking at with the frog embryos was really the CDC2 product, which is a kinase. We're going to call it the CDC2 kinase combined with a cyclin. So whatever was getting the frogs to mature or go through those divisions was actually really a combination of two proteins, a cyclin and a CDC2 kinase. So to look at the diagram here, you would have a cyclin that's increasing in concentration, and you have your CDC2 kinase, and then they combined together to make this complex. Okay. Now the CDC2 kinase, they're going to refer to them as cyclin-dependent kinases in general, or CDK for short. So CDC2 is going to be a specific kind of cyclin-dependent kinase. And what does that mean? It means that the CDK is a kinase. It's going to phosphorylate, but it depends on binding with a cyclin to make it active. Okay, so these cyclin-dependent kinases were then found, based on this research, to be the positive drivers for the cell cycle. In other words, they were important in triggering the cell to go to the next stage. So looking at the checkpoints, there's going to be 3 of them. One of them is going to be right here. So this is that cell cycle. This is interphase right here from the start of the yellow here to this. So here you have a new cell, and that cell begins to go through growth. and then it'll either stay in G1 and then we'll call it G0, or it may progress and go on to S, right? This is a major checkpoint here because it's very important. If we're going to start making DNA, you cannot stop. The next checkpoint is right here. And this checkpoint is going to make sure that we've copied our DNA correctly before we go into mitosis. And then the spindle checkpoint is going to be the one where we make sure everything is good before you start separating sister chromatids. We've kind of already mentioned these here. They are all, every one of these checkpoints is going to be controlled by those cyclin-dependent kinase enzymes. So you're going to need those cyclin-dependent kinase enzymes to trigger moving on to the next point here. So here's your first one. It's the G1S checkpoint. Sometimes it's called the start or the restriction point. This is the major one there. And this is where a cell is really going to decide whether it's going to go through a division or not. This is also going to be the primary point where the cell responds to external signals from, say, maybe other kinds of molecules that are in the area. Then you have the G2M checkpoint between G2 and going into mitosis. And here, we're going to commit to going through mitosis itself. And here we want to make sure that DNA has been replicated properly. Okay? Otherwise, we don't progress. And then here we have the late metaphase or the spindle checkpoint. We want to make sure, because you're going to get to that metaphase stage, you want to make sure that all your chromosomes are lined up on that metaphase plate and they're attached to the spindle fibers before you progress. Otherwise, you're going to end up not separating the sister chromatids correctly, and then each cell will not get the right number of chromosomes. So the cyclin-dependent kinases are going to be the ones that drive that cell cycle, right? So we've already mentioned that they're called kinases because they phosphorylate. So these are going to be enzymes that phosphorylate other proteins. These other proteins are involved in controlling that cell cycle. So you've got to activate them. The cyclin-dependent kinases are going to be those primary mechanisms for that. They are going to partner with or bind with cyclins at different points in the cell cycle. Okay. Now, for many years, it was the view that it was the cyclins that drove this cell cycle. So the cyclin binded with, if the cyclin was present, and we know that the cyclins increase and then decrease, so they are made and then they're degraded at certain points in the cell cycle. So if they're present, then they bind with the cyclin-dependent kinase itself and then allow the cell to progress through the next stage. Okay. So it was thought that that was the sole control of it. But it's now become clear that the cyclin-dependent kinase itself is also controlled by phosphorylation. So the cyclin-dependent kinase needs to be phosphorylated too. So think about this. Here's your cyclin molecules. So say you have cyclin being produced and you have an increase just before we start progressing to the next stage. If it's present, then it should bind with a cyclin-dependent kinase. Okay, so it binds. But just because they bind doesn't mean that that's it. It also depends on whether or not the cyclin-dependent kinase itself is phosphorylated. So it has to be phosphorylated too. If it's not phosphorylated correctly, then it doesn't matter that the two are bounded together. Okay, so there's another means of controlling this situation here. Okay, so think about this. Here you have your cyclin-dependent kinase product binding with your cyclin at this checkpoint. So here we're going to say this one's a G1 cyclin. And then over here you have, it's the same kinase, cyclin-dependent kinase, but a different cyclin, the M cyclin or mitotic cyclin. And then over here you have your anaphase promoting complex, which is cyclin-dependent kinase with cyclin as well there at those checkpoints. So the CDK or cyclin-dependent kinase, it's a protein kinase, and the way it's going to work is it's going to activate other proteins that are actually more directly responsible for getting the cell to progress. So we might think of the cyclin-dependent kinase as switching on other proteins that are involved in getting the cell to progress through the cell cycle, right? So we've got to do that and phosphorylate it. So we can see that in the model up here. So we have our cyclin-dependent kinase. The cyclin-dependent kinase is phosphorylated, and then that complex itself goes to an inactive protein, some protein that's a different protein that's involved in getting the cell to progress, or a series of other proteins. And so this cyclin-dependent kinase is going to go and phosphorylate the target protein that's inactive. So right now it's inactive, and the cyclin-dependent kinase is going to transfer that phosphate on there, phosphorylating it, and that activates it. that protein then will go on and do other things that get the cell to go through the cell cycle. So that's the way it works there, right? So if we're looking at the one that we've been studying that's going to promote that frog egg and any other cell now, it was initially observed within the frog in production of frog eggs. So they call it maturation promoting factor. But because we see this common to all cells that are going through the cell cycle, it was renamed to the M phase promoting factor for mitosis, right? That same one, the one that is between the G2 and the M phase checkpoint, right? So when we look at the cyclin-dependent kinase itself, and we come back to it right here and how it's phosphorylated, it turns out that it depends where that phosphate is bound to whether or not the entire complex becomes active to go activate those other proteins, right? So that's what this next part here is talking about. So the activity of the cyclin-dependent kinase itself is also controlled by the pattern of its own phosphorylation, because it also has to be phosphorylated itself. Okay, so we relate it to the diagram that you actually do see in your book. Here's the way this diagram, which I got from another source, that cyclin is drawn this way, but here's the cyclin from the diagram in your book. Okay, and you can see that this cyclin is bound to a CDK or cyclin-dependent kinase, and you can see that this cyclin-dependent kinase, like the one that was over here, this is the way it's drawn in the book, actually has two different locations where it can be phosphorylated. If the phosphate is bound in one location, this would be inhibitory, and that means this cyclin-dependent kinase is not going to go activate the other proteins, right? And that means we stop the process. We pause the cell cycle, okay? If it binds in another location, that's going to be active. So that's why they've symbolized this by saying red here for stop and here green for go. So if it's red, that would inactivate, and then that means we would not be able to activate those other target proteins, right? However, if the conditions are right, then we phosphorylate in a different place, and then that allows the process to continue. Okay, so when we look at specificity, we focus in, we go back to where we were studying the frog eggs and whether or not we progress through mitosis. We're looking at the M phase promoting factor. So it's once thought that that M phase promoting factor was controlled solely by the level of the M phase specific cyclins. Remember, there's the amount of cyclin that's in there, and that's it. So you remember what's going on at this checkpoint is we've just finished making copies of your DNA, and they need to have been done correctly. Okay, so it turns out that it's not just that the M phase cyclin is present, right? Not that this molecule is present, and it's not that it's present and bound to your cyclin-dependent kinase. It also depends on where that kinase is phosphorylated, and where the kinase is phosphorylated depends on whether or not the DNA was copied correctly or not, right? So if you have bad DNA during the copying, okay, there's going to be a complex pathway that's going to cause the phosphorylation on this complex to be more in that location, and that stops the process, so we don't progress. We don't want to go divide and give two new cells damaged DNA. So we got to pause at that point and stop and decide whether or not we can repair it or destroy the cell itself. Okay, so not just the cyclin, not just having the cyclin-dependent kinase, but it also depends on if the conditions are right. So here we have a mechanism. If the conditions are right, we're going to phosphorylate in the green location. If the conditions are not right, things went bad, we're going to phosphorylate and stop the process. Okay, so now we're going to move on to the checkpoint where we check to see if the chromosomes all lined up correctly on the metaphase plate. So that's the spindle checkpoint. And this one involves another one of those cyclin-dependent kinases, but they give it a name, anaphase promoting complex, and it also goes by another name called cyclosome, which is for that C there. So this is going to be at the spindle checkpoint, and we want to make sure that all our chromosomes are on that metaphase plate, that imaginary plane where they're supposed to line up, and that the tension is correct in the microtubules, the kinetochore microtubules that are connected to each of the sister chromatids, right? So if all of that checks out, then the anaphase promoting complex is activated, and what it does is going to trigger anaphase from occurring but it does so indirectly. And what's it going to do? Well, it turns out that there's another set of a pair of proteins in there. It's a securin separase complex, and the separase, that means enzyme. And while the separase is bound to securin, the separase is inactive, right? So what's going to happen here is this APC, when the conditions are right and everything, all the chromosomes are lined up correctly, this APC is going to go and mark the securin right here, and it's going to tag it in a whole separate other set of processes that tags it for destruction by other enzymes called proteases. Okay. And so the protease is going to go and destroy this protein, and when it destroys it, it frees the separase, and that causes it to become active. And what is the separase going to do? It's going to go and destroy the cohesin. And what is the cohesin? Recall from the earlier section we're talking about chromosomes. The sister chromatids that were copied are held together by that protein called cohesin. And so if they're all lined up, all your chromosomes are lined up on the metaphase plate under tension from those kinetochore microtubules, they're being pulled on. If we unleash the separase, a bunch of separase molecules, not just one molecule, it's a bunch of them, and you unleash them all at once, they're all going to be there breaking down that cohesin, and once it's broken down, then the sister chromatids all begin pulling apart all at the same time. So it's an indirect mechanism, as all of these are, right? So, but they all require these cyclin-dependent kinases to go and activate other proteins. In this case, we're activating separase. Okay, so when it comes to eukaryotic cells, eukaryotic organisms that are multicellular, things are even more complex. So a lot of the stuff we were talking about was discovered in yeast, and you understand yeast, and that provides a good model for understanding more complex situations. And one big difference here is that in yeast, they only had one kind of cyclin-dependent kinase at your checkpoint for the G1S checkpoint, and the same thing at the G2M checkpoint. There was only one kind of cyclin-dependent kinase. And you went back and you studied that diagram more closely here. You can see that it says CDC2 inactive and then active, and then here it says CDC2 here. In this diagram looks a little different. This one says CDC or CDK2, same thing, CDK, and this one says CDK1. So you have two different kinds of cyclin-dependent kinases. You're also going to have different kinds of cyclins. Cyclin E, cyclin B, and so on. So what is this? When you have more of these cyclin-dependent kinases, that provides even more control. When you're a multicellular organism, the cells need to be very coordinated with respect to each other. We don't want different cells are going to divide at different rates, and you have to exercise exactly more control. The yeasts, they're only responding to basically one external cue, and it's involved in mating. So yeasts only need one basic cyclin-dependent kinase because there's not a whole lot of controls needed for the yeast. But in multicellular organisms, you need greater control. Okay, so when we look at external cues, signaling in a multicellular organism, those are generally called growth factors. There's many kinds of growth factors. The first kind of growth factor that is going to trigger the cell to progress through the cell cycle is called platelet-derived growth factor, and this PDGF. And this one is found in your blood, and if you damage your, say you get a cut in your skin, this platelet-derived growth factor is going to signal cells in your skin to begin to divide to repair the wound. Okay, and this growth factor, like all growth factors, are going to override the controls that would inhibit cell division. So what the growth factors are going to do is, these are external molecules that bind to a receptor, and then that sends a signal in and says, we need to progress through there. Right now you're on pause, but we need to progress and cause the cell to divide. The platelet-derived growth factor is going to act on a specific kind of receptor called an RTK, which stands for receptor tyrosine kinase. So itself is a kinase. And this molecule that's located in the plasma membrane, it's a receptor, is going to actually when it binds, it's going to change shape and then cause a series of other reactions to occur within the cell. Whenever you have an external signal, let's say there's a receptor there, here's your cell, this is your plasma membrane, and that other signal molecule comes and binds here. That signal molecule doesn't go in and send the signal, and instead it transduces the signal inwards. Generally, that's called signal transduction in biology. So this RTK, receptor tyrosine kinase, is a specific kind of signal transduction that generates a pathway of phosphorylations that occur there. So this will be better understood if we look at it on our diagram here. This is your plasma membrane, your phospholipid bilayer, your nucleus is right there. And here are your receptors here, and this is just some kind of growth factor. So let's say this is our receptor tyrosine kinase, and it's a kinase because we're going to phosphorylate, right, other molecules. So what happens here is if your growth factors are present, they're very specific for very specific receptors, they're going to create a series of phosphorylations, and those phosphorylations are going to go on and activate different kinds of proteins. Now, there's all kinds of abbreviations here. Don't focus on memorizing this. I'm not going to test you specifically on the RTK pathway here, but you should recognize that you're going to see a series of phosphorylation there, okay, that we're going to activate on one molecule, then that one molecule is going to activate our first kinase, and so this is going to phosphorylate another molecule here, which is itself is a kinase, activating it, and you can see it's become phosphorylated, and then this serves as a kinase, and then eventually you get to a molecule that's inactive that'll become active, and this one right here is that MAP kinase that was, there's MAP kinases are a family of kinases. This one is a specific one called ERK, but the point here is we've activated it. This MAP kinase then enters the nucleus and goes and phosphorylates another protein that is involved in control. And right now, this protein right now is saying, we have the brakes. We've put the brakes on progressing through the cell cycle. We're not going to be going through any division, cell division. But when you go and you phosphorylate this particular molecule here, the red one, it was bonded with another molecule in there. And that molecule, the E2F here, is inactive right now. As long as this RB protein is bound to it, it's inactive. I'll tell you what that protein is in a bit, but here comes that MAP kinase, phosphorylates it, and gets it to fall off. Okay, so there's your free regulator protein, and that frees up this molecule here, which is a protein, and it's a specific kind of protein generally called a transcription factor. And transcription is the process when you go from DNA to RNA. And let's say we're going to make a messenger RNA. That messenger RNA is going to take the directions to a ribosome to make a protein. Okay. In this case, what protein might we be making? Well, we might be making those cyclins. Remember, cyclins are going to be the ones that go up and that are going to help the cell progress through the cell cycle. That cyclin needs to bind to a cyclin-dependent kinase. That cyclin-dependent kinase needs to be phosphorylated correctly, and then we can progress, but you need the cyclin there to begin with, right? So this is an example of how we can take that signal in and then get proteins made that we need to get the cell to go through that, continue through the cell cycle. So in this section, we're going to be looking at cancer, and your learning outcomes here are going to be to distinguish between proto-oncogenes and tumor suppressor genes. And then you're going to look at a specific protein called a p53 protein, which is involved in suppressing uncontrolled cell growth. So after we finish the section on how we control the cell cycle so that we don't have uncontrolled cell division, we define cancer then as being uncontrolled or unrestrained cell division. So cancer then would represent a failure in the control of the cell cycle. And we see that there's a connection to certain kinds of genes. There's two kinds of genes that when these genes become mutated can result in uncontrolled cell growth and the formation of tumors and cancer. One group is called proto-oncogenes, and oncos is cancer. So if you're an oncologist, then you would be one who is studying and curing cancer. But a proto-oncogene would be, these are genes that normally help a cell progress through the cell cycle to go through cell division. And they're a normal part of that control. You need to go through the cell cycle, and then you also need to pause it when it's important to pause the cell cycle. So those would be called tumor suppressor genes. To use an analogy that the book said, if you're in a car, these are like the gas pedal. You hit the gas so you can go. These are like the brakes. Okay, and you need both of these working properly to control the cell cycle. You need cell division, but you don't want uncontrolled cell division. So looking at proto-oncogenes, these are normal genes that can become oncogenes when they're mutated. And oncogenes cause cancer, right? So these proto-oncogenes might be genes that code for proteins that are involved in, say, they're involved in growth factor receptors or any of that signal transduction pathway that causes the cells to go through cell division. So we were looking at the RTK pathway, the receptor tyrosine kinase pathway. This is that same image from there. So imagine any one of these proteins that are along the way, starting with the receptor there, to any one of these kinases. If any one of those becomes mutated in such a way that they're on a permanent on condition, on switch, then they're going to constantly cause this cascade of process to occur that gets the cell to go through cell division. Okay, so that's why they refer to it as a mutation that causes them to be permanently on. So let's say we mutate these receptors where you no longer need the signal from the outside. The receptor, the RTK receptor, is permanently on, so it's constantly causing this cascade of phosphorylations to occur. That would be bad. And so that's an example of, these would be proto-oncogenes. These would be genes that code for these kinds of proteins that are involved in this transduction pathways. So we call that signal transduction because you bring the signal in and then causes that cascade of reactions that activate the cell to go into cell division. Now, the important thing to remember for these proto-oncogenes is you only need one bad copy. So you get two copies whenever a new individual is produced through sexual reproduction. You get two copies of every gene because you get one full set of chromosomes from the paternal and maternal side. They come together and you get two copies of every gene. All you need is one of those, the genes that are for any one of these proteins to go bad, just one copy, and then that can cause uncontrolled cell division. Okay, so that's an important thing to remember. So while one good gene is producing good protein, the other one is producing the bad protein, and all you need is one copy, and it's uncontrolled cell division. Then you have tumor suppressor genes, and these are going to be different. As long as you have one good copy, then those proteins are suppressing or preventing the cell from dividing. Okay, so one example is that gene that is for making the P53 protein. Okay, there's another one, the RB or retinoblastoma, which is actually a form of cancer. It is identified in a certain kind of cancer, but itself, the protein, is actually a tumor suppressor. Now here, unlike the proto-oncogenes, you have to have both copies go bad. Okay? So as long as you have one good copy, it's still doing the suppressing. Okay, so that's important to remember for tumor suppressors. Okay, so you basically have to lose function of both copies in order for certain kinds of cancers to develop there. Now, one of those first tumor suppressors to be identified was the one that causes a cancer called retinoblastoma, and it's a cancer in the eye. And so you're predisposed for this if you have a parent that's carrying a bad copy. During embryonic development, when the retina is forming, the cells inside the eye and the back that are sensitive to light, as they go through division, a mutation could happen in the other good copy, and then you get retinoblastoma. So predisposing means that you're a higher risk of getting that cancer early in that development of the eye. So it's a cancer of the eye. But the gene itself, the protein, is also important in other cells in controlling the cell cycle as well. So if we take a look at the RB gene for retinoblastoma, as long as you have one good copy, we saw where it was. It was located in here. Okay? That was that RB gene we were talking about in the earlier section here. So you can see here you have your RTK transduction pathway, the receptor tyrosine kinase 1. And so you have, say, a growth factor here. And you go through the series of phosphorylations of these different kinases to eventually you get the MAP kinase that comes in and phosphorylates the RB, detaching it. And that's the important part here, because as long as it's detached, it's preventing that protein that's colored in yellow here, which could be a transcription factor that gets a gene to be transcribed that would produce either cyclins or cyclin-dependent kinases, and both are important for going through the cell cycle. Well, imagine if you have a bad, both copies of the RB gene, the one that can cause retinoblastoma. If both are bad, they're not going to be attaching to and suppressing the transcription factor. And so that would cause those cyclins to be constantly being produced. And so you would get uncontrolled cell division in that case, right? So this one's talking about the role it plays, this particular gene, in the cancer called retinoblastoma. If you inherit one bad copy in the early embryo and then through thousands of cell divisions there, if the other copy goes bad because of a mutation when it's copied, then both copies are bad and you're not making normal RB protein. And then that leads to cancer in the eye. So what does the RB protein do itself? It doesn't itself cause retinoblastoma. It's an important tumor suppressor gene in that it suppresses cell division. And what it does is integrates those signals that are related to those growth factors, right? And so the growth factor coming from out here, it integrates the signaling. Here's the integration right here. Okay? And so it's basically the role of that protein there is to suppress the transcription factor from transcribing and then translating proteins like cyclins and cyclin-dependent kinases. So here, its role is to slow or stop the production of cyclin and cyclin-dependent kinases. Remember, those are those complexes that are involved in those checkpoints, right? So we want to stop at a checkpoint. And if you have that RB gene, you've lost control over that. Another one is that P53, the name of the gene, it produces a P53 protein. And this P53 protein plays a key role at the G1S checkpoint. So it's the G1S actually checkpoint here. And what the P53 protein does is it monitors the DNA. Make sure that the DNA is in good shape, the integrity of it. If your DNA becomes damaged because of sunlight, ultraviolet light, or carcinogen, some kind of a mutanogen that causes a mutation in the DNA, this P53 plays a role in stopping progression past the checkpoint so that it can activate enzymes that go and repair the DNA. And if the DNA can't be repaired, then the P53 is going to direct other proteins that are going to cause the cell to basically implode on itself and kill itself. And this process is called apoptosis for programmed cell death. So the cell gets programmed to die, and that way the cell doesn't go and divide and make more bad copies that can ultimately result later on in cancer. So this prevents the multiplication of mutated cells. So if this P53 is damaged, then you don't have that exercise, that control. So we can see here, if we have a normal P53 gene and you have damaged DNA, that P53 protein, whatever caused the damage, could be radiation, ultraviolet, heat, chemicals. That P53 protein is not itself going to go and repair the DNA. What it's going to do is going to activate DNA repair enzymes that go in and try to repair. If the repair is good, then the cell is going to be allowed to continue once you repair it, to progress past the checkpoint, go through S, then G1, and then make new cells, and then you have a good line of cells. If the repair is no good, then we're going to activate enzymes that are going to cause the programmed cell death, so the line of cells dies, so you can't repair. Now imagine if you have a mutation and you have bad P53. So you have abnormal P53, it can no longer do its job. And so we can no longer activate repair enzymes. And so if the DNA mutates, the mutation is now going to show itself in new lines of cells through cell division. And if you have further mutation, not necessarily the P53, but some of those other genes, okay, say some of the other proto-oncogenes, then eventually that line of cells can turn into cancer. But you can't stop the process from progressing because you have your bad P53. So in summary, you have your proto-oncogenes that help push the cell cycle forward, and you have your tumor suppressors like the RB protein that help put the brakes on that process overall. Now, for the proto-oncogenes, you're only going to need one bad copy, and that causes the cell to continue to go through the cell cycle and the cells to divide. For tumor suppressors, you would need both copies to become mutated, so two bad copies of the gene. Now, this does summarize here. Here you have your tumor suppressors, and here you have those involved in that growth factor receptor and that cascade of kinases that cause the cell to progress through the cell cycle. So when we look at some of the proto-oncogenes, when you have a mutation in the growth factor receptor, this is found in many cases in breast cancers. When you have that one of those protein kinases involved in the transduction pathway, that RAS protein is going to be mutated in 20 to 30% of all cancers. There's another kinase that we saw that's part of that phosphorylation cascade that's active in 2 to 5% of cancers. You don't have to go memorize these just to show that these have been discovered to be mutated in these kinds of proteins. When we look at the retinoblastoma protein, the RB protein is mutated in 40% of cancers, and the one that's found in many of the cancers is the mutation of the P53. Now really they're discovering that in different cancers you have a different set of genes that have been mutated. So it's not just always just one gene that's mutated. It's usually certain types of genes in certain types of cancers. So that's understanding those genes that are mutated might help develop better therapies for treating these cancers in the future.